My name is Amita Kaundal, and I am Assistant Professor in the Department of Plants, Soils and Climate in the College of Agriculture and Applied Science, Utah State University, Leuven, United States. Today I am going to talk about my PhD student Vishal Singh's work exploring transcriptional memory landscape for salinity stress in maize seedlings. As we know that world population continue to increase, which demand an increase in food production. However, the globally arable land per capita is shrinking. Furthermore, the climate-induced um, environmental changes, environmental stresses have reduced crop production. Among them, abiotic stress are the top one impacting crop production, which is evident from this graph. Soil salinity is such a kind type of abiotic stress that drastically impact food security. Soil salinity is a problem that is predicted to increase over time, which is evident from this picture that which is predicted for 2031 to 2060, how world soil is going to be more salty with time. In such condition, we need to develop salinity stress resilient crops. For this study, we chose maize because maize is the most produced crop globally with production of 1.2 billion tons, followed by wheat and rice. In 2024, the United States was the largest producer of maize. However, maize growth is negatively affected if soil salinity exceed 4 decimum per meter. The most affected growth stages in maize are germination and seedling stages. As we know, the plant they are able to uh, perceive the stress and then able to respond to it. So when plants are exposed to different stresses by either abiotic or biotic, they can sense the presence of these stresses by these receptors or sensors present on their plasma membrane. And perception of these stresses induce a cascade of signal transduction which activate different protein kinases and transcriptional factors. And these transcription factor then ultimately induce the expression of stress responsive gene. So plants has this ability to remember the stress or have this memory to remember this stress and prepare the plant for future stresses. And this process is called priming. So plants' ability to remember this stress memory, which we want to study in this um, experiment, where we have exposed V73 inbred lines for different stress episode of 8 hours with 40 hours recovery phases to the saline solution of 12 decimum per meter in hydroponic uh, situation. At the end of each stress or beginning of the stress one, we collected um, uh, physiological data and then we collected leaf and root tissues for mRNA sequencing. The same set of experiment also done without any salt, stress, um, and samples were collected at each step as a control. The 9 different physiological parameter has been collected. Uh, Relative water content, assimilation rate, transpiration rate, stomatal conductance, electron transport rate, steady state chlorophyll fluorescence, quantum efficiency of photosynthetic electron transport under light through photosystem 2, non-photochemical quenching, and carbon dioxide inside the leaf. The relative water content data revealed that plants were able to retain the relative water content after stress 2, and they were completely able to retain relative water content after stress 3. The other physiological observations, we found similar patterns in stomatal conductance, electron transport rate, assimilation rate, and quantum efficiency, where plant were able to um, maintain these parameter after stress 2 and stress 3. The correlation between various physiological traits across different stages and recovery phases revealed that relative water content showed a weak but significant correlation with quantum efficiency and electron transport rate, and a big negative relationship with non-photochemical quenching and uh, carbon dioxide inside the leaf. The transpiration rate showed a strong positive correlation with the stomatal conductance. The transcriptomic analysis revealed that plants, these samples, they group different samples grouped together based on their gene expression values. So you can see that all root samples, including salt and recovery phases, they are grouped together. However, the leaf recovery phases and stress phase leaf samples, they grouped into two different groups. We also found that the differential expressed genes were found more in leaf as compared to the roots in all three stress episodes. Um, Based on this data, we classified differential expressed gene into non-stress memory genes, stress memory genes, and delayed response genes. So, if the genes they are induced or repressed in each subsequent stress episode, they are considered as memory gene, and if they are not induced in consecutive stresses, uh, they are called non-memory gene. So based on this data, we found four genes in leaf which are induced in all stress phases. However, we couldn't find any gene in roots. We found 320 genes in the leaves which are induced in stress 1 and stress 2, and 21 genes which are in roots which are induced in stress 1 and stress 2. Similarly, we found 9 genes which are genes in leaf which are repressed in stress 1, 2, and 3, and 336 genes which are repressed in stress 1 and 2. However, we couldn't find any gene expressed in roots for all three stress phases, and 69 genes we found in root which are expressed to all two stress phases. We found some delayed response genes. That means these genes they are not expressed in first stage, but or repressed in first stage, but they are expressed, induced, or repressed in stress 2 or stress 3 or only stress 3 phases. So altogether, we found 746 genes in leaf and root showing a stress memory response. We found that 4.44% these SMG or stress memory genes in leaves are transcription factors, and 5.36% of these genes in roots are transcriptional factors. And these genes belong to different transcriptional families like Burkitt, MIB, HDGIP, or ERF. Here are some representative of expression pattern for SMG in leaf, where you can see these genes which are induced in all stress phases. They are also induced in the recovery phases also. Or if they are repressed in stress phases, they are also repressed in the recovery phases. Similar pattern, expression pattern of SMG in roots were found, and these are just some of the representative genes where you can see the induction of these gene in roots on salt. They also induced in the recovery phase also. We also identified the gene modules by weight co-expression network analysis that by WGCNA, where we found that module pink and purple showed a significant positive correlation with relative water content, and in module green and black showed a high positive correlation with NPQ. The Go enrichment analysis revealed the involvement of photosynthetic pathway or gene involved in photosynthesis and gene involved in metabolic processes like abiotic stress, photosynthetic light harvesting in photosystem 1. These genes are play a significant role in salinity stress. And based on this data, we have shortlisted top 10 genes which are associated with salinity stress in maize. So few of these genes are in differentially expressed in leaf. Some of them are differentially expressed in roots only, and three of these genes, aquaporin, chaperin, DNAJ, putative MIB, DNA binding domain superfamily genes, which are expressed differentially expressed in leaf and roots both. So based on the from this list of 10 genes, we selected 4 of them to see their expression patterns to validate their expression by QRT-PCR, and we found that these SMG showed similar expression as displayed by RNA-seq data. So to summarize this work, we found that there are 746 genes in leaf and roots showing a stress memory response. A total of 37 genes were found to be transcriptional factors. Stomatal conductance of water, relative electron transport rate, assimilation rate, and quantum efficiency increased during stress 2 and stress 3. A declining number of DAG with subsequent stress episode was found. Most of the top Go annotations were for the biological processes linked to the photosynthetic mechanism and regulation of metabolic processes. We also shortlisted top 10 genes which are which of them belong to the category MRE11, VHLH transcription factor, AP2, ERE, BP transcription factor, or methyl binding domains. The co-expression network analysis showed a coordinated expression of genes from different pathways to ultimately regulate physiological response. So now our next step, we are still analyzing this data with different combination permutation. But in future, we are going to investigate the gene knockouts for these shortlisted SMGs for their potential role in stress memory. And we are also planning to study the epigenetic aspect of salinity stress memory. With this, I will end my talk and thank you so much for your time. If you have any question, I am happy to answer them. Thank you so much.